So, well, thank you so much, Chris, for such a nice introduction and also for being such a wonderful host. So both Chris and Robin have been amazing hosts, and I really feel very honored for having been invited to speak at the Warren Wagner series, and feel very lucky to have arrived this week, because I see on Sunday it was a little cold, and I see all the spring flowers coming out, and it's been really fun just throughout the week seeing more flowers coming out on the way. I also feel like sort of like a kid in a candy store talking to people in the department. It's been really amazing, and it's really fun, amazing to see like how diverse the biology department is. I was also really pleased to talk to people in the geology department, and it's been really, really fun to be here. So, as you probably saw from my title, I'm going to be talking today about the origin and evolution of the neotropical biota, and I'm going to use some examples from the plant family Bignoniaceae, which, as Chris says, is my favorite group, and I've been working on this for a while now. So we all know that the origin of the patterns of species diversity and the causes of present-day distribution patterns have really intrigued evolutionary ecologists, biogeographers, and ecologists for a long time. And in the neotropics in particular, several hypotheses have been proposed to explain current patterns of species diversity. So we have, for example, hypotheses that go back to the 1800s, like the riverine barrier hypothesis proposed by Wallace, and then the refuge hypothesis that was really popular in the 1970s, also the ecological gradient hypothesis, among many others. However, it's still unbelievable that the exact factors that have shaped neotropical distribution patterns are still actually poorly understood. Although we know that both historical and ecological factors have actually played key roles in shaped current species distribution patterns, and so if we really want to understand the origin and evolution of the neotropical biota, we really need to have an integrative and cross-disciplinary approach combining data from systematics, phylogeny, ecology, evolution, biogeography, geology, population genetics, among others. Of course, in order to really understand the origin and evolution of biotas, we want to adopt like a taxonomic analysis, and we want to have data from many different groups, and that's really the ideal world. However, before we actually have robust data from many different groups, we actually need to have in-depth analysis of individual lineages. And I started off with the Bignoniaceae and thought I would work on this for a couple years and would know everything about them and would switch into other groups, but I've been working on them for 20 years now, and there's still a lot to know about them. So this is my model group, but actually this is a key component of the neotropical flora. It includes around 840 species and 90 genera. There's a variety of habits, so trees, shrubs, and lianas. They are distributed around the world, but they're predominantly neotropical, and their species occur in all major ecological zones, from occurring in both wet and dry habitats. They are also very diverse morphologically and ecologically. They're a really important component of the tropical flora. So even though this is a case study, I think I really have a great model system here, and I hope to convince you till the end of the talk that this is really a great group. So before we start any kind of study, I mean, systematics is really the basis. I was originally trained as a plant systematist and then moved into evolutionary biology and ecology. But this entails so lots of field work. I was amazed just to visit the herbarium here and seeing the incredible size of the herbarium, and really exciting to see now the museum coming in. And the value of these collections is just unbelievable. And so I did a lot of field work throughout the neotropics, and also my students collecting in many different places. We are also very lucky to work on the Bignoniaceae because this was the group that Al Gentry, which is one of the most prominent botanists and who was also the world's leading expert on the Bignoniaceae. And Al worked on this group for a long time and compiled and collected all over the place. Anyone who knew him knew his capacity of just collecting things, and he left a really wonderful collection at the Missouri Botanical Garden, which has over 100,000 specimens of Bignoniaceae alone. Very well curated collection. And this has also been an invaluable resource for all my research during all these years. So of course, during all these years, so lots of field work, lots of herbarium work, working on many different floras, and these kinds of work have been really instrumental in getting us to really know in detail the species that occur in different places. And this has allowed us to produce a world checklist of the Bignoniaceae. And we currently recognize approximately 840 species, although more than 4,000 species have been described in this group. We also have a fairly good a molecular phylogeny for the family, and this has allowed us to recognize 8 major clades, which now are recognized as tribes. There are 2 genera that are still unplaced, and 1 group is now recognized as a separate family, the Schlegeliaceae. And within the past years, I have actually focused most of my research in the tribe Bignoni, which includes half of the species in the tribe, in the family. So these are approximately 400 species and 21 genera. And this was the group that needed most of the work, most work in the Bignoniaceae when I started. And this is where I have focused most of my efforts during all these years. So this is really the most abundant and diverse clade of lianas in several neotropical ecosystems. We were talking about this right before the seminar. So we have many different liana families that are very important in different places, but these are usually independent lineages within each family. And this is like the most important clade, most diverse and abundant in most neotropical forests. They occur in all major neotropical biomes. And we currently also have a really good phylogeny for the tribe. This was an initial study in which we sampled 1/4 of the species. So here's 100 out of 400 species in Bignoni. And this was really critical to establish a new classification for the group in which only monophyletic clades that are recognizable by unique morphological features are recognized. This new generic classification led to lots of changes in lots of changes in the classification of the group. And as we can see, the classification was very varied a lot over the years, and we sort of needed a more stable system. It involved like approximately 200 new combinations, approximately 80 new lectotypes, and a lot of taxonomic work to get this sorted. And so in this synopsis, we were able to establish sort of a general framework for studies in the group with good diagnosis for at the generic level, and with a quick synopsis for each species and good distribution maps for every single species. So this was sort of a first step, just outlining what are the genera and what are the species recognized. And now my students are working on a bunch of taxonomic revisions of all of these 21 generic level clades that I showed you before. And we've progressed substantially in all of these taxonomic revisions. These are really so species level accounts of every single species with detailed distribution maps and illustrations, and also descriptions for every species. And we're doing all the monographic work using as basis new phylogenies for the group, and it's sort of one informs the other. So phylogenetic data provides a lot of interesting information for the monograph, so we're able to sometimes identify cryptic species. We also are able to test the monophyly of taxa, and at the same time the monographic work also brings important information for the phylogeny. So this is just to show sort of the background and where we stand with starting with Bignoniaceae research. So we have a very solid systematic background. We know what our units are. We know the distribution of taxa, and we have a good understanding of phylogenetic relationships within the group. So after doing all these studies and getting a really good understanding of patterns of morphological variation in the group, we wanted to go to the next step and start to understand patterns of morphological evolution in the group. So the group has lots of really interesting morphology. So for instance, in the stems of these lianas, we have a very unusual pattern of phloem growth. And what happens is we have a regular phloem over here that is separated by a variant phloem. So we have these different patterns in the group, we wanted to first look at the pattern of evolution of phloem types in the group. One interesting thing is that each of these wood anatomical types evolved a single time. So while most of the other morphological features are really homoplastic, these wood anatomical traits are really stable in the group. And one thing that is really interesting is that all species start with the common development, so they all start with a continuous phloem, and then we have the variant portions that are in equidistant portions within the regular phloem, and they start to produce more phloem and less xylem in these portions. And then we have some species that stop their development at this stage, and there are other species that go one step forward, and then they start again to develop these phloem wedges again in equidistant portions of the regular phloem, producing these phloem wedges in these different portions. And then some species stop their development at this stage, and others go forward and go into the multiple dissected to this next step. So actually they all have a very common evolutionary pattern, and they just, some taxa, some clades go one step further, which actually suggests a recapitulatory history. And then we also wanted to understand, so what is happening at the cell level, and what is happening in the in the evolution of the individual cells within this group. So here we can see very clearly, so the regular phloem, the variant phloem, with more phloem being produced to the outside and less xylem to the inside, and the cambium separating the phloem and xylem, right? So when we look at the patterns of evolution of different cell types, what we found is that in the variant phloem, we actually have sieve tube elements that are much larger, and we also have a lot more fiber. And this actually suggests an increase in conduction and mechanical support. And while we look at the regular phloem, at the cells present in the regular phloem, we actually find smaller sieve tubes, larger ray cells, much more parenchyma, and less fiber, which actually suggests a reduction in conduction and higher storage capacity. Another trait that always made us really interested also were the tendrils. So of course for lianas, this unusual wood anatomy and also the tendrils are really key features for their, even for their survival. So the first thing we did was to look at the general pattern of evolution of tendrils in this group. So we have tendrils in Bignoniaceae are modified leaflets, and we have tendrils that are simple. In some other cases, these tendrils are biparted or parted 3 times at the extreme, or multi-part, like parted multiple times. So we first wanted to see the general pattern of evolution, and we saw that actually the ancestral condition are trifid tendrils, and then we have several reductions to simple tendrils, some reversals to trifid tendrils, and evolution of multifid tendrils, and which is actually the opposite of what Darwin actually hypothesized, because he hypothesized a single evolutionary path from simple to bifid, trifid, and multifid. But actually we start with trifid, reduce to simple, and so forth. And so we wanted to understand the ontogenetic pattern and how these tendrils were formed. So after reconstructing the evolution of tendrils, we then went on to study the ontogeny to see how the different tendrils are formed. One thing that was really interesting was that when we studied tendrils, species with trifid tendrils, and which are nested within lineages whose immediate ancestral also have trifid tendrils, very early on in development, they would start as triphid and would just continue to develop as triphid. And when we examined species with simple tendrils that were nested in lineages whose immediate ancestor had simple tendrils, these species would also, from the very early on, develop as simple tendrils. But actually, when we looked at species that had triphid tendrils, but whose immediate ancestor had simple tendrils, what we found is that they would actually start to develop as triphid tendrils, and later on in development, there would be a reduction of these triphid tendrils, and they would end up as simple tendrils. And this exact same thing, species with simple tendrils that were nested in clades with triphid tendril ancestors, they would start to develop as triphid and then have a reduction. So it was really interesting to see that they still carried some information, and then during their development, we would recover those patterns of the immediate ancestor in their final outcome. We were then also interested in seeing which were the genes that were involved in these tendril changes. And since tendrils are actually modified leaflets, we looked at some of the genes that are associated with changes in leaf partition. So these are the shoot meristemless, leafy floricala, and fantastica. And these are genes that are directly associated with leaf partition. And then when we looked at the patterns of expression of simple tendrils, triphid tendrils, or multifid tendrils, we actually found exactly what we find in modified leaves and higher expression patterns of these three genes that are associated with division in triphid and multifid tendrils exactly at the moment in development in which the tendrils were dividing. So just confirming that they are really modified leaves and function as leaves. The group also has quite a bit of lots of extrafloral nectaries that are associated with different insect-plant interactions. So for example, we have lots of mite domatia that are associated with different mite types. We also have species that have hollow stems that house ants, sort of like a Liana cecropia or something. We also have species with lots of extrafloral nectaries between the petioles, extrafloral nectaries in the profiles of the axillary buds, and lots of nectaries in different parts. So we wanted to understand the evolutionary pattern of these nectaries and also see whether there was an adaptive role. Because usually people would just say, oh, you know, you have extrafloral nectaries, they secrete sugar, sugar attracts ants. These ants come to feed on sugar, and that impacts fitness, and it's an adaptation, a protection against herbivores. So the first step in order to understand whether the extrafloral nectaries were really an adaptation was to first see what were these extrafloral nectaries. And we found out that they're actually trichomes. They're glandular trichomes, and there are multiple evolutions of different kinds of these glandular trichomes in different clades. We then went on to see whether they really had a defensive role and whether they were really playing a role in defending these plants against the attack of herbivores, which are all the steps associated with the test of adaptation hypotheses, of course. And then when we went on to actually test the adaptive role and the selective regime on these nectaries. So one thing that was really interesting is that we found within species variation in these nectaries. So for example, you can see this is the same species, and you can see that some individuals have a lot of nectaries, while other individuals have only few nectaries. And actually, some of the individuals with very few nectaries, they attract a different composition of different ant community, while the individuals with more nectaries attract different types of ants. And so we did a population level study in which we sampled multiple populations within the Espinhaço range, and we found that the individuals that had lots of nectaries did have a protective role and protected them against the attack of herbivores, and these species actually, and that had an impact on the fitness. while the individuals with only few nectaries these nectaries had no protective role, and they were largely attacked by herbivores. So essentially, within a species, there's variation in nectar, and you have some populations that sort of work as hotspots with lots of nectar being produced, lots of ants that protect and having an adaptive role, while other populations within the same species, there's no protective role. So this actually matches perfectly the expectations of GMT, which is the geographic mosaic theory of coevolution of Thompson, in which you have this geographic mosaic of variation within a species with some individuals. So really, nectar is really having a protective role and not in others. Another character that always called our attention are flowers, of course, which are very diverse and variable in this group. And the first step was sort of just understand general pattern of evolution in discrete morphological characters associated with flower morphology. So we selected a bunch of discrete characters, and we used a likelihood approach to conduct ancestral state character reconstruction using all the individual floral traits. And one of the things we wanted to see was first the order of the events and when did individual flower traits evolve. We also wanted to see if there was a correlation between the evolution of particular floral traits that are thought to be associated with individual floral syndromes. And indeed, there was a strong correlation, and different traits associated with floral syndromes evolved together. In some instances, we saw sort of a punctuated change from one sort of syndrome to another, and in others, we would see mixed morphologies that would be would have intermediate character stages, and that would lead to the subsequent flora type. The next step then was to look at continuous characters. So we looked at a series of continuous characters. These were characters that were associated with some of the external worlds, which are associated with calling the attention of pollinators, and also characters associated with more internal worlds, which are actually the reproductive characters, so the gynoecium and the androecium. And when we looked at the evolution of these continuous flower traits, which were 16 quantitative traits, we found higher phylogenetic signal than expected under a Brownian mode of evolution in traits associated with calyx and corolla, actually suggesting phylogenetic conservatism. So this was suggesting that traits associated with calyx and corollas were more constant than expected by chance under each clade. So essentially within clade, you had flowers that had similar sizes. On the other hand, when we looked at the internal worlds, we found lower phylogenetic signal than expected by chance under a Brownian mode of evolution, which indicated high homoplasy. So essentially you have within a clade flowers that look the same size, but you have a lot of variation in androecium and gynoecium, which is actually suggests that this is the mechanism that might be leading to to isolation between different species. We were then interested, so we were first looking at the evolution of individual traits, so just discrete and then just continuous traits. And then we wanted to look at the overall pattern of evolution and to see whether there was phenotypic integration among all the different traits. So instead of looking at each individual one, seeing whether flowers were evolving in an integrated fashion or not. And what we found was that, first of all, flowers have very similar patterns of variance and covariance among traits. The evolution of flower morphology has occurred through the evolution of magnitudes of correlation among traits, which suggests that stabilizing selection has actually played a really important role in phenotypic integration, leading to the long-term stasis of covariance patterns underlying diversification. So essentially what this means is that these different traits are evolving in a correlated fashion, and you have this long-term stasis. So essentially, this long-term stasis in phenotypic integration is actually what allows us to recognize particular patterns of floral morphologies as distinct attributes of different botanical families. So there is this integration, and there is stasis in the evolution of those traits, and this is why we look at a flower and we say, oh, that's a Bignoniaceae flower, or that's a Lecithidaceae flower. It's just because there is this correlation and stasis in the patterns of phenotypic integration. So a good understanding of morphological evolution is actually also critical for the study of community assembly. So more specifically, this is really a good understanding of morphology is really key to determining the actual ecological forces, such as competition or environmental filtering, that actually explain the patterns of phylogenetic structure in a given community. So we then wanted to take a look at how these morphological features were influencing community assembly. We wanted to further understand general patterns of community assembly in this group. So we know that environmental filtering actually leads to communities with species that are more morphologically similar than expected by chance. So all the traits more morphologically similar than expected by chance. While competition leads to communities that contain species that are more different than expected by chance. And so using these principles, what we wanted to do here was to look at the effect of phylogeny, environment, and morphology on different communities of Bignoni. More specifically, we really wanted to evaluate the role of environmental filtering and competition mediated by pollinators for Bignoni community assembly. So what we did was to use Gentry's transects. So there's a large database with Gentry's transects, and we are really lucky because the Bignoniaceae are extremely well identified in those transects. And we also complemented the 89 transects in which species of Bignoni were recorded with 5 additional transects that were compiled by Fernando Martins, a professor at Unicamp in Brazil, for the Atlantic Forest of Brazil and Cerrado, leading to a total of 94 transects localities in total. We then also used this information to extract data for 5 environmental variables from the 1904 communities. We used ArcMap, and the environmental variables that we looked at were mainly associated with temperature and precipitation. So like the mean amplitude of monthly temperature, annual amplitude in mean monthly temperature, annual precipitation, and distribution of precipitation throughout the year. And then we used our time-calibrated tree as base to look at the phylogenetic component. We then also used all the discrete morphological features that we had already studied before and the quantitative morphological traits for which we had a good understanding of the patterns of evolution. So the first thing we wanted to do was to assess the impact of phylogeny on species co-occurrences. We did this from a species perspective and also from a community perspective. So from a species perspective, we tested for pairwise associations between the species co-occurrences and phylogenetic distance using patristic distance. And we actually found that phylogenetic distance was not significantly related to co-occurrence. And then when we redid the analysis, but then considering a community perspective, we also found no phylogenetic structure in the communities that were sampled. So overall, so the communities of Bignoni are not phylogenetically structured. There is, the close relatives do not co-occur more or less than expected by chance. We then wanted to look at abiotic preferences and habitat specialization. to see if there was habitat specialization in this group. And in order to do that, we used a PCA to reduce all 5 variables that we measured for each community to a small number of independent variables. And we quantified the abiotic preferences by calculating the convex hull for each species. So essentially, the convex hull, which is this thing here, gives us a measure of the abiotic preferences for the individual species. So like a species with a convex hull like that would have very narrow abiotic preferences, while a species like that would have broad abiotic preferences. We then created a null distribution for the convex hull, and we tested whether species exhibit ecological specialization by checking whether the convex hull area differed from the new distribution for the actual convex hull. So here's what we got. So here's our convex hull area, which is our measure of abiotic variation. And here's species occurrence. So what we have here is, in gray, all these little dots, the null distribution of the convex hull. And in black, we have the mean distribution of the convex hull. And in white, these are the actual observed values of the convex hull. And as you can see, a lot of the observed values are over here. And statistically, we found that species of Bignoni actually occupied lower convex hulls, so they actually occupy a narrower range of abiotic conditions than expected by chance. So this means that the species of Bignoni, they actually tend to occupy a limited portion of the potential convex hull space that is produced by their abiotic, predicted by their abiotic variables. Also means that most species are actually specialized to a restricted set of abiotic conditions, which suggest that species distribution are actually driven by environmental conditions, with habitat filtering representing a key driver of the community assembly in Bignoni. Finally, we wanted to look at the effect of floral morphology on species co-occurrences. We looked from a species perspective and from a community perspective. We found that floral divergence was not significantly related to co-occurrence when we looked from a species perspective. And when we looked from a community perspective, we found also that floral diversity observed within communities did not differ from the null expectation that communities are assembled randomly. So this means that the distribution of the floral diversity in Bignoni did not differ from the null expectations that communities are assembled randomly in terms of their flower morphology, indicating that competition for pollinators is not driving community assembly in this group. So after taking a look at community assembly, we wanted to go to the next step, which is actually to look at broader patterns and biogeographical patterns in the group. So first step was to time calibrate our phylogeny. So we used 2 fossils as calibration points and an uncorrelated log normal model of rate evolution to infer trees and associated divergence times. And we used some standard ancestral state reconstructions to try to infer the biogeographic history of the group. So here's our time-calibrated tree for the tri-Bignoni. And what we see here is that the group originated in the Atlantic forest of Brazil at approximately 50 million years old. And then we have a major occupation of Amazonia and major diversification in Amazonia. We then have several returns to the Atlantic forest, 1, 2 here, 3, 4, 5, and 6. 6 of these events are at around 10 million years, and 3 of those are at around 10 million years, and The other 3 are at around 20 million years. When we look at the history of South America at the time, we actually see that around 20 million years, there were some potential for connection between Amazonia and the Atlantic forest, which would explain the connections from Amazonia to Atlantic forest at around 20 million years. And also some recent research has shown that apparently there was a connection between Amazonia and the Atlantic forest between 5 to 9 million years, while there was a connection through the southern portions of South America during the last 5 million years in the last glacial maximum. So probably the other connections, half of the connections were through northern portions of South America, which is in agreement with these forest corridors that were present at around that time. We also see major occupation of dry areas and large diversification at around 30 million years, which is also around the time when a lot of these dry forests were forming over here. So we had a broad picture of what was happening in terms of biogeographic history of Bignoni. The next step then is to reconstruct detailed phylogenies for all of these groups and then address individual specific biogeographical, test specific biogeographic hypotheses using each of these lineages in a very complete sampling. So remember that this tree has 1/4 of the species, so 100 out of the 400 currently recognized. And now, so we're in the process of reconstructing all of these phylogeny. Some are finished and others are on the way, and looking at the biogeographic history within those clades. So just to get a little sample of the things that we are finding, this is the genus Anemopegma. The genus, it's around 30 million years old. And what we see is, so when we go to the most recent common ancestor, we see that it was broadly distributed between C and D. So it was in western South America and Amazonia. We then see a split with a lineage remaining in Amazonia and diversifying in C, and another one, here, and then another one diversifying in western South America. We then see an expansion into B, which are the dry areas, and then over here, and substantial diversification within B. So while when we look at Bignoni, we see origin in the Atlantic forest and a movement more towards north, when we look at the individual clades, it's exactly the other way around, right? So we see origin in western South America and Amazonia in a movement towards the south, although there are multiple movements, it's not just a single event. So we have a similar pattern with Bignonia and the group. So when we look at the most recent common ancestor, it was in western South America. Then we have an expansion into Amazonia, substantial diversification and movement in Amazonia, subsequently occupying dry areas and diversifying in this region. But of course, if we want to understand the history of neotropical taxa, we also need to look at the rest of the world and see the influence of different regions to what we see in the neotropics. So the next step then was to get the whole phylogeny of all the Bignoniaceae and do a broad biogeographical study. So we used the published phylogeny of the Bignoniaceae, and we were able to compile 56 different fossils. So this is work that is still in progress, and just got back from Asia on a trip that we are trying to get all these fossils on loan and examining all these different fossils. And anyone trying to do this knows that it's a lot of work. So what I'm going to show you is some preliminary results with the data that I have thus far, but we're still working on getting better estimates from these fossils and improving our analysis, also including a higher number of Asian taxa in our published phylogeny. So we use sort of the standard methodology that we had already used in other studies, and we coded our species using this distribution coding proposed by Colt. So here's our time-calibrated tree of the Bignoniaceae. And of course, it's hard to see here, so what I'm going to do is just to walk you through the independent clades and show you really quick what we see within these different lineages. So the whole family goes back to around 60 million years. And then one of the first lineage that diversified in this group is Jacaranda, which is approximately 10 million years old. The group is restricted to the neotropics, especially South American. So there's not much to see here in Jacaranda, given that this is a very broad analysis. Next lineage that diversified is an Andean clade that is actually approximately 40 million years old. It has only a few species. Bignoniaceae is not very abundant or diverse in the Andes. The next lineage that diversified is Tecomi. And what we see here is that this group was originally in South America. We then see an expansion into Africa and another one into Central America and North America. We then have a connection with Asia, which is actually a common pattern that we see in different groups, a disjunction between North America and Southeast Asia. And the group diversified substantially in Australia and also in H over here in the Himalayas. And what is interesting here is that we see, so this one species, Campsidium valdivianum, which is actually from South America, but is nested in this clade from Australia. But this is actually not so surprising, given that this is a very well documented pattern, with species disjunctions between Eastern Australia and Western South America. And when we look at the geological history of this region, South America was actually still connected to Australia through Antarctica up to 30 million years ago, which is actually a very short time. And around this time, so Antarctica was warmer, so this would provide a path for this connection. So we don't need to infer a very long distance dispersal, because there was this land connecting this region. We have another clade, which is another disjunct like North America and Australasia, which is Catalpa, that lots of you know. And so Catalpa and Chilopsis, this is actually hybridized. This is sister to actually a group from South China, Malaysia, and the Pacific. Then we have the Paleotropical clade. And the most recent common ancestor was either in Madagascar or in Africa. And we see several patterns that repeat within this clade with African lineages that are sister to lineages that are in Madagascar, and African clades that are sister to Asian lineages. See again another clade with major diversification in Madagascar, lineage from Madagascar, sister to Africa, sister to an Asian clade with some other African taxa in it. The last two lineages, there's not much to see in it, because these are groups that are endemic to Central America and South America. This is the Tabebuia alliance. Tabebuia is particularly diverse in the Antilles. And but the group was originally in South America, reached Central America, and diversified extensively. And Bignoni, we've seen a much more detailed analysis that I showed before with much more detailed coding, but what we see here is that the group diversified extensively in South America. So overall, what we see is quite a bit of movement between South America and Central America, at least 17 different events. One big connection through Africa, some probably through long distance dispersal, but at a time in which these land masses were much closer. We then see movement between Africa and Madagascar, connections through Madagascar and India, but also at a time in which these land masses were much closer. Some extensive diversification in the Himalayas, quite a bit of movement in Australia. Two disjunct taxa that imply long distance dispersal, but again at a time in which these land masses were closer through North America and Asia, and one one South American taxa that is nested in the Asian one, which is that connection through Australia. So these are sort of the general patterns that we see in Bignoni and in Bignoniaceae. And Bignoniaceae, as I said, is a really important group for the neotropics and for the tropical flora as a whole. But of course, as I said at the very beginning, if we really want to understand assembly of biotas, we really need to do cross-disciplinary analyses and cross-taxonomic analyses. And this is actually what we are doing in this project, which is a NSF Dimensions of Biodiversity in partnership with FAPESP. It's a biota project. So we have this very large project funded, which includes many different collaborators from many different institutions. So here are some of our key collaborators. You can see Chris here. He's another PI on this grant. We have all the geology team and some of the primate people working on butterflies. So in this project, the first thing we'd like to do is to really characterize Amazonian biodiversity. So we really want to first establish how is biodiversity spatially distributed across Amazonia, how are species distribution organized into patterns of endemism, and what are the biotic and abiotic environmental associations with these diversity patterns. So for that, we're using 4 major focal taxa, so plants, primates, butterflies, and birds. And after looking at the patterns within each of these groups, we want to answer some more general questions, which are like, so first, are the patterns of endemism congruent across groups? Or do these patterns relate to the environmental history of Amazonia? And the second component of the project, so after characterizing the organisms that are in Amazonia, is to then also reconstruct the phylogenetic and phylogeographic history of selected taxa. And with that, we mostly want to understand what has been the evolutionary history of the Amazonian biota and how was it generated. So for that, we've selected several clades of nymphalidae and different butterflies. We have 6 different genera of primates, 8 different clades of birds, and 2 main plant groups, Bignoniaceae, which is the key liana group, and Lecithidaceae, which is one of the key tree groups in Amazonia for this phylogenetic and phylogeographic reconstruction of taxa. The third component of the project is a characterization of the geological and environmental history of Amazonia. And I think this has been like particularly interesting for us biologists in this project, having this chance of interacting with geologists. And essentially what we want to do with this component of the project is to understand what has been the history of environmental change across Amazonia, also understand when did the Amazonian river drainage actually form, and what was the Amazonian landscape like before the Amazon River formed, and how did it change after the river formation. So in terms of the river drainage, there are 2 competing hypotheses that are, so some believe that the river drainage formed between 8 to 10 million years during the upper Miocene. And another hypothesis is that actually formed in the upper Pliocene or Pleistocene, so less than 2.5 million years ago. So geologists are trying to gather a lot of new data to try to answer these specific questions, which will be really important for our biogeographical studies, in which we want to combine the data from the geological studies with the data, the organismal data from our selected focal taxa to really reconstruct the biogeographic history of Amazonian taxa and understand what has been the temporal and biogeographical history of the Amazonian biodiversity. This is a very fortunate time because we have this Amazonian project going on, and at the same time there's another project that aims to do exactly the same thing with the Atlantic forest. Most people are involved in both projects, so we have the PIs on this project are Anna Carnaval and Cristina Miyake. So these projects always have like one main PI in the US and one in Brazil and several other PIs in the US. And we are all also involved in the Atlantic Forest Project. So it's really an exciting time to have these projects looking at these integrative questions at the same time. So just want to thank all the funding agencies that have provided funds for these projects, and also want to thank some of my students, so these are the people who have been involved in the taxonomic and phylogenetic work. These are the people who have been involved in the more evolutionary components of this project, doing the evolution of different traits, like Mariani has done all the tendril evolution, Marcelo the wood anatomy, Susanna the flower morphology evolution, Flavio has done his thesis on the evolution of mite domatia and associated mites, Anselmo looking at the insect-plant interactions, and André Rochelli looking at the evolution of seed dispersal in this group. And here's some of the new people, like these are two new postdocs that are now taking the lab into next generation sequencing, and some students that are now starting to look at phylogeographic components, which is a relatively new component to my lab, and so we'll need a lot of collaboration on that, but it's an exciting new frontier. So I'd be happy to answer any questions. Robin?
It's interesting that the diversity of the Bignoniaceae seems to be so incredibly recent, at least on your tree it is. And I'm wondering, given that some of the earlier things still have these beautiful floral attractants, do you have any thoughts on what might have stimulated such diversity in that one group?
So you mean the current diversity? So the Bignoniaceae evolved at around like 40 million, well, 50 million years ago. And then, but when we looked at the age of species, they are much more recent. So do you mean the species age or the whole
Yeah, I guess when
tribe?
you went through your tree carefully, that was one of the last groups that you
Oh,
showed
yeah.
just how it was, I couldn't quite...
Yeah, no, but it's actually 50 million years. So yeah, I was just showing different clades, but when you look at the ages of clades, it's actually pretty old, 50 million years.
One of the most diverse of the groups?
Yes, yeah.
So again,
yeah, what are the drivers? So a lot of these traits associated with the liana habit, so the wood anatomy and tendrils and things like that have been hypothesized to have been key innovations that led to the diversification of this group. Unfortunately, we have a single evolution of these traits in Bignoniaceae. So in order to test whether these things are really key innovations, we'd need to do like a broad-scale angiosperm phylogeny analysis and looking at other groups of lianas that have similar traits to see whether they were also drivers of diversification in these groups.
In general, so within the Bignoniaceae, the Bignoniaceae, you did find evidence for habitat specialization.
Yep.
That at a comparative level, if you were able to compare that to the tree diversity in the neotropical trees or the Amazon, did you find a comparable level of habitat diversity, or are lianas much more generalist in their...
That's a very good question, and we don't know. But it's a really good question. It would be really interesting to see that. Yep. Yep.
Well, I have to say that having known Herb Wagner, he would have thoroughly enjoyed this.
Ah, thank you.
Just looking at your photo there, you did show, when you showed flowers, a fair diversity of flower morphology. Yeah. Is there much specialization for pollinators at the lower levels here, or
yeah.
general?
So we have 5 different pollinators in this group. So we have bees, hawk moths, butterflies, bats. What am I missing? Bees, and birds, of course, hummingbirds. That's it, yeah. One thing that is really interesting, as I showed the pattern of flower evolution, so in some instances we see a direct change from one sort of pollination syndrome to the next, and in others we see mixed morphologies that are intermediate. So one thing we're trying to do now with an undergraduate is also looking at species pairs and seeing, for example, sometimes we have a hawk moth species that is sister to a hummingbird's pollinated species, and we're trying to look at the pollinators and also seeing if we, in the hawk moth species, if we also get visits from these other taxa, which would imply that they're sort of maintaining some of the ancestral traits. We're also looking, for example, if we have one hawk moth species that is within a clade that is pollinated by a different pollinator. So we're trying to see, we see that there's a diversity of things that visit, and we're seeing whether the diverse community of pollinators that visit actually reflect some of the ancestral traits that were maintained in these flower morphologies. So it will be really nice to have more empirical data in the pollination studies to know the pollinators for such certainty, with certainty. Yeah.
To follow along with that question, I was thinking about the result of how calyx and corolla were very conserved while the sexual whorls were very labile. What about flower color?
Very labile. Yeah. So we have multiple evolutions of all the different flower colors, which, yeah, great point. It would complement, so as another mechanism isolating as a barrier between pollinators apart from the internal floral whorls. Exactly. Yeah. Yeah. Yep.
About the phloem anatomy, do you see any sort of trade-offs between like high amounts of conductance versus structural stability? And is there any correlation between those phloem anatomies and the habitat? Or they somehow specialize for the particular habitat that they're growing?
Yeah. So there are, within Bignoni, there are also some shrubs. So there's a single evolution of the liana habit, and there are reversals to shrubs. And even within the shrubs, we still see some of the wood anatomy, but in some cases, it's completely lost. So they maintain, so as I said, all species, they start with like a continuous phloem. They start developing like that, and then you have the phloem wedges, the 4 and then the 8. So in some of the shrubs, we see a reversal in that wood anatomy. Well, they just stop the development at that stage. And what was the other question?
What is it linked to their habitat? Like, do fast growers have a certain type of phloem anatomy, and would that help them in a particular sort of habitat?
That's a really interesting point. And there's, so Marcelo Paci, who's the student who led all that work on the phloem, he in collaboration with some other people are really doing now a lot of functional analyses and trying to get into the actual functional associated with these. So we'll probably have this answer to this question in a couple years. Yep.
As part of what that group is testing for, the different phloem types for 8 multi-wedges confer different levels of what we might call engineering strength, elasticity, flexibility to the vine.
Yeah, exactly. So the higher the phloem wedges, so there's more flexible tissue, and it gives more flexibility to these vines. Exactly. And also, it has another big advantage, which is if you have a continuous phloem and it breaks, the plant dies. But with these phloem wedges, it sort of prevents the phloem from breaking. It has, in the plant, there's no embolism associated with that. So it really seems to be a very key component for these lianas. Within Bignoniaceae, there are multiple evolutions of the liana habit, and only the clade that has the specialized wood anatomy and tendrils was very successful and diversified extensively. All other lineages only have a few species.
I'd like to comment with maybe one more.
Yep.
So you mentioned earlier about the amount of nectaries being associated with possibly the rivers. So how did you assess fitness and conclude that it wasn't as necessary?
Yeah. So we collected fruits. We did the whole step. So we went into the field and we saw how many fruits were, we counted in the field how many fruits were generated, and we actually waited to see until these individuals actually germinated. And actually, after some of these germinated, we brought them back into the lab, and now we're doing a lot of like greenhouse experiments and crossing them to see patterns of heritability in those. So we're doing common garden experiments to actually understand the heritability of those traits. So we have a big greenhouse with all the plants that grew out of these field season, and we're growing them now.
Right. Let's end here. If anybody who wants to come up and talk to Lúcia right now can. And let's thank Lúcia for a fantastic talk.
No, thank you.